So you can make a lot of things. I think my slide is not going to work here, but one of the things that you find when you grow these cells, much to your surprise, is you look down the microscope and they start beating. And you're making cardiac myocytes spontaneously, without trying. The cells just form. So that gives you some idea how limited our control is over these cells at the moment. The cool thing is, what also forms are spontaneously retinal pigment epithelial cells. And again, that's been exploited such that you don't have to do anything. That cocktail of growth factors that I told you about is not needed. You just let these cells grow. The black ones turn out to be retinal pigment epithelial cells. And they really do appear to be authentic, bona fide retinal pigment epithelial cells. Okay? Our job is a little more difficult because that doesn't happen with retina spontaneously. We have to do that stepwise. Now, I'll take you through a little bit of the data here to show you that we really can make cells that look like and work like photoreceptor cells. So this staining that you see here in this panel is an antibody stain that indicates to us that these cells are making rods and cones following this differentiation procedure. And the bottom panel shows that they work, that you can stimulate them like you would a photoreceptor, in this case with glutamate, and they fire. So we start with these cells here at day 0 that don't work. They're not neurons. They're undifferentiated neural stem cells, IPS cells, induced pluripotent stem cells. After 33 days, they turn into a neuron that works. This is just a high-power view of what some of these retinal cells look like after they're transplanted. And so a photoreceptor cell, a rod or a cone, has two ends. Okay, it has an end that captures light, and it has an end that sends that information onto the rest of your nervous system. Okay, so it's a polarized cell with two ends. Most neurons are different. They have two ends that make connections. A dendrite and an axon, we call them. So photoreceptors are unique in the body in that they have two bits, a light capturing part and a part that sends that information on. And so to study these cells in a transplant, we want to make sure that they make both of those parts because they're both essential to function. And so we study them with a lot of different methods. But principally, it's immunocytochemistry, it's called. It's staining, using antibodies to find out if these cells are making the right proteins. So in this case, we're looking at a protein called rod outer segment membrane protein 1. This tells us they're making the light capturing cells that are needed. These are called outer segments and have been really a challenge for people to get to grow from a transplanted stem cell. So now I would say there's 4 or 5 groups around the world that have shown that you can transplant stem cells from a variety of sources and they grow outer segments after transplantation. And that means they have the capacity to capture light. Doesn't mean they're going to work, but it means they have the capacity to do that.